Welcome to the Evolutionary Hologenomics Podcast. In this episode, we have Raphael Eisenhofer. That's a very good start. I probably just butchered your name there, but you'll let me know. He's a postdoc working with us here at the Center for Evolutionary Hologenomics. So welcome, Raphael. Good evening. It's 8:30 in the morning here in Copenhagen, but you are not in Copenhagen. So where are you? What do you do? And what time is it over there?
Well, thanks, Anna. I'm currently in Australia. In a city called Adelaide, which people don't really know much about. People, when they think of Australia, they think of, Brisbane, Melbourne, Sydney. Those are the kind of major cities. But Adelaide is the capital of South Australia, which is one of the states. So it's kind of, if you think of Australia as that kind of big continent, it's kind of in the center. This is like nice little section right in the middle called South Australia. So that's where I'm currently at. And it's 6 p.m. currently here.
6 p.m. And it's the summer, right? Because here it's December. I guess we're all heading towards Christmas for those who celebrate Christmas. But over here it's snow time, and over there not so much.
No, it's a pretty hot time actually. We had a nice 35 Celsius day yesterday, which is a pretty moderate summer day. We can get temperatures above 40. I think last year we actually had like one of the records for the city of like 45.8 degrees Celsius or something, which is just, it's like hell. Yeah.
Yes, that sounds very, very hot. I'm just gonna change the view here. Okay, now we are both on the screen. So I understand that you work with these very interesting animals called marsupials. Are they only in Australia? Because this is where, if people think of kangaroos and so on, we think Australia. Do they only live there?
Oh, that's a good question. It's a very good question. If you think of marsupial, most people would think, as you said, kangaroo or maybe koala, the cute fuzzy bear that lives in the tree. But there's actually, in fact, quite a diversity of marsupials living in South America. But by far, I think the most diversity of marsupials is actually from Australia in itself. And particularly the diversity of form that we have in Australia is quite unlike anything else. A lot of the South American marsupials, they all look quite similar. Although I'm sure if you ask someone who works on South American marsupials, they're like, no, definitely not. But when you compare this to Australia, where we have the kangaroo, a really unique, distinct form, we have things like wombats, these stocky little quadrupeds, all the way to possums and tiny little rodent-looking things. So it's really quite an amazing diversity of marsupials we have in Australia. But there are marsupials outside. And I guess if you live in North America, you probably have the opossum. There's a species of opossum that's quite big over there. I've not been to North America before, but I think they're around quite a bit over there, scavenging and stuff.
Yep. And actually, I heard, is this true or not? Or, what does the scientific evidence say? That actually they did not originate in Australia, although now there are more of them there and more famous and more known and more species, I guess. But where do they come from? Where was the first ancestor found? Do you know?
I don't know the exact details. I think it probably relates to when the continents were one, and then they obviously split apart. I'm not sure exactly. I think people maybe think it's kind of the South America region, perhaps, but you'd have to ask someone else for that. But all I know is that within Australia, in the context of Australia, when it separated from the continents, the marsupials were there, and somehow they kind of took over the continent. And as a result, the Australian marsupials, they diversified, that they evolved in all these different forms and shapes that we have now in Australia, and occupied all those different niches within there. So until recently, when European mammals came in, but we can talk about that maybe a bit later.
Yes, and I have more questions, and I can see that you have a really nice picture here, so I will ask you more about the different groups. But I just realized that I went straight in about marsupials, but the aim of this podcast is also to kind of inform people and keep everyone in the loop, and maybe have people learning new things. So maybe we should start with the basics. What is a marsupial, and what's the difference between, for example, a marsupial in the animals with placenta and so on?
Yeah, no, it's a very good question. So maybe we should first ask the question, what is a mammal? Because a marsupial is a type of mammal. We know that we are mammals, but in the general sense, there are kind of three major groups of mammals. We have the monotremes, which I think is Latin or Greek for single whole. So that they basically, how we have specialized genitalia and, an anus, monotremes just have a single whole. And that's also true for marsupials as well. So they have what's called a cloaca, which is kind of like, I think, birds and various other animals do. But monotremes are very unusual. You've probably heard of one of them. A very popular example also comes from Australia, called the platypus, which is quite an unusual critter. It's kind of like a bird and kind of like a mammal. I think there's stories of when Europeans first were told about this. They're like, oh, that must be some kind of elaborate ruse to take the Mickey out of us. But they are actually true. They're very interesting because they lay eggs. Monotremes, which is quite interesting. Whereas, obviously we know that we give birth to live young, etc. So that's a monotreme. There's also the placenta, the placental mammals. So that includes us. So that's this kind of group here, which is quite a huge diversity of mammals that people are probably more aware of, things like tigers, bears, elephants, etc. And they're distinct that they give birth to quite developed young. Whereas marsupials, on the other hand, they give birth to very mature young, which I'll talk about in the next slide. And maybe I'll just add one little thing. So these are all mammals, right? But what is a mammal then? Do you know, Anna? What a mammal is?
A long time ago, we learned, I don't know, because it's very interesting as well, because biologists learn these things, even children at school at some point, I think we learn this, but then new knowledge and new evidence appears, which is a good thing. So all the books I have, genetics books and things, they're so outdated compared to now. So what we learned at school a long time ago, the main feature was that mammals carry their young and give birth to them, and then they feed them with milk. And that was kind of the main feature of being mammals.
Yeah. I think, and particularly the milk one is, I think, the big one there, because, the name mammal, I think it's, mammary, which, milk, etc. So that's a real key feature for mammals, which is not to say that some other animals don't produce milk. I think there's some snakes and stuff, but that's one of the many vagaries of biology. There's always exceptions to the rule. You think, there's a rule for absolutely everything. A mammal must be this and that, and nothing else can have that. But there's always some little obscure animal that breaks the rule for its, particular group, which is always interesting. So, given that we're talking about marsupials, I guess we'll focus a little bit more on them now. What we know about them, in terms of some of their differences and whatnot. One key thing is they shared a common ancestor with us. So, our ancestors, we shared a common ancestor with them all the way back here, which, if we look at this phylogenetic tree, which is, a tree of life that we've created using sequencing data, we see that we share a common ancestor with marsupials about 170 million years ago. That's our best estimate of when we did this. So since then, basically, those two different groups have kind of gone off on their own different evolutionary trajectory and diversified and not shared genes with each other. So this is quite an extraordinary period of time, right? 170 million years ago. I mean, geez, like a year is a big time for me, but 170 million years is just crazy. So this is just give you an idea of, how different we are to them in terms of time, and how much time has elapsed between us and them.
Yeah, so that's very interesting, because it is a long time ago, and I guess the more time something has to evolve, then the more different and specialized it can become. Do you have any idea of, you or scientists in general, of what was this common ancestor and what it kind of looked like? Because now mammals and marsupials, and even within each group, there is so much variety. Do you know kind of what it looked like?
I think the best guesses are probably something pretty small, something kind of like, I don't know, I don't want to say a rodent, but like, something pretty small and mobile. I guess, one thing to consider as well is, around this time, the world was dominated by dinosaurs, big scary reptiles, ruling the planet essentially. And so, there probably weren't many big mammals or any big mammals at that time, right? Because those niches were already occupied by these big dinosaurs. So I think people think, for the most part, that they were small, they're kind of hiding around, opportunistic scavenging, etc. And then really it's kind of when I think the dinosaurs went extinct around 60-ish million years ago. That's when you start to see a big diversification of all these different forms, right? And that's when you look at this tree here. I think this is the KT boundary here. This is the fancy term for that. That's when dinosaurs kind of die, and then all these other new different forms have an opportunity to evolve.
Right, because now they could come out of their burrow a little bit, or wherever rock they were hiding under, and they could just occupy all the different niches and can go on land and trees and whatnot. So that makes sense once the dinosaurs were out of the picture. So marsupials, this is what you've been working on and in the past how many years?
So probably the past couple of years now, I think. So I finished my PhD in 2018, which was on a completely different topic on ancient DNA and microbes from humans, from ancient DNA. And then I guess I kind of fell in love with the biodiversity in Australia. Like, it's kind of hard not to, because it's just so astonishing and amazing. That I thought, well, why don't I study this instead? There's not many people working in this area in particular, and I'd love to also kind of eventually move this research into helping these mammals as well, because we're learning a lot now at the CEH, and the CEH is doing a lot of really great work about looking at how these microbes interact with us to influence our health, and what we can do to change that, and hopefully give us better outcomes, etc. So extending that to mammals would be a long-term goal of mine.
Nice. And so I don't know, well, I do know what's coming next, but so we talked about the pouch, and what's it for?
Yeah, so this is one of the key features of marsupials, and I think the Latin or Greek for marsupial is pouch. And so essentially, I mentioned before that, placental mammals like us, we give birth to live young that are pretty developed. Humans, I think, are a bit of an exception, right? Like, you think of a baby and
useless.
Quite a number of years until it can do anything, right? But, this example, like deer, for example, I think that they can walk or run, a day or so after being born. So much more developed. But in marsupials, they're born extremely young. So we're talking between 3 to 6 weeks of pregnancy, which, I'm sure if you're a mum, that's probably a nice thing. I don't have experience being pregnant, but being pregnant for 9 months might be quite a challenge. But with marsupials, 3 to 6 weeks, and the marsupial comes out. The joey is what it's called, and it crawls into the pouch. And so this pouch is this, I guess, fold of skin that can be quite like a sac, a pouch, or it can be quite, not necessarily a pouch at all, just kind of like a flat bit of muscle that will kind of wrap around the young. And here's some examples of some pouches. So this is a yellow-footed rock wallaby, so kind of like a type of kangaroo. And you can see the little pouch young sticking its head out there. And this is not a two-headed wombat. This is actually joey in the pouch of a wombat. And what's actually kind of interesting is where the pouches are kind of differ based on the animal. So what's quite interesting with wombats is their pouches are kind of facing backwards for the most part. And some people think that the reason for this is because, wombats like to burrow a lot. They dig a lot of, dirt. If the pouch is facing upwards, then all that dirt would kind of go into the pouch, and that could be a challenge for the young. And so hopefully by, facing it backwards, there's going to be less, dirt going into the pouch and hurting the joey.
Yeah, so for the people who are just listening to the podcast and not watching, we are looking at 2 very cute pictures of a wallaby with its joey in the pouch, and then we have a very funny, also very cute, but very funny looking wombat. To me, this picture is very funny because it does look like it's giving birth or something, but obviously it's too big and hairy. It's already much older than when it was actually born or when it came out. But it's just a very funny picture because I think the image we have when we think of marsupials is the kangaroo jumping around, and you can see, obviously, because also it jumps on 2 legs and the wombat uses 4 legs, but we see, mom and baby sitting upright, whereas the wombat is a very different image that we get. It's very interesting. Yeah. And do we know about, well, I guess it's with everything, some animals evolve and they have some features and others don't, and then they all live in the various niches. But the pouch is obviously a very good thing for protection. But so why don't, for example, rabbits and other things have pouch? Do we know that? Is it the niche or the pressure, or what? Do you know what was the what might have been the factors that led to some animals developing a pouch and others not, when they live in similar environments or have similar predators or challenges or so on?
Yeah, it's a really good question. I think there's a few ideas around. So, I mean, I guess probably the most basic is that, sometimes once evolution gets going, it's very hard to kind of go back, so say you start to have pouch young, it's very hard to go back and just want to have, a full, very developed birth. So yeah, sometimes, things kind of get stuck in those little evolutionary trajectories. Some people hypothesize that in the context of Australia as the continent, giving birth to pouch young very early can be beneficial when the climate is moving around a lot, when you have extremes like droughts and stuff like that. The idea being that you quite easily terminate that birth, that you're not putting a huge amount of energy into that pregnancy. You're given young to something very small, and if the environment isn't great for you at the moment, you can just get rid of it. It's not a problem. That's, I think, one idea. But yeah, it's a very interesting question.
Yeah, and I have many questions now that I have somebody from Australia working on marsupials. The questions are endless. So you mentioned that they are only pregnant for 3 to 6 weeks. Do you know which species are have the shortest and longest gestation?
Not off the top of my head, but my guess would be probably the smaller marsupials might have the shorter, gestation times than maybe the larger ones. That'd be my educated guess.
Yeah, that makes sense. And then they have to crawl all the way up to, well, I say up, but in the wombat, I don't really know what they do. But my question is, how do they know? Do they have claws, first of all? How do they get up there or down there? And how do they know where to go? Is there some kind of chemical cues, or they're born into the pouch? No, they're not born into the pouch. They're born and then they go up to the pouch. So how do they know where to go?
Yeah, no, I mean, that's a very good question. I'm not really sure. I mean, so they're born quite, while they are born very young, certain parts of them are quite developed. So like their legs, particularly their front legs, are very developed. So this is an example of a really young pouch young from a rock wallaby. So the same image that I showed you before. And this is about 1 to 5 weeks old. And so you can see it has made it into the pouch already. It's moved there. And it is still pretty developed, like look at these arms. It could still manage to crawl its way up there. This is it on the teat there. And you also notice that this is another little random aside, but some marsupials have different numbers of teats in their pouches, which is quite interesting. And those different teats have different kind of milk, formula essentially for when the joey grows up, which is pretty interesting.
Right, so they just move up, I guess, right, instead of having just all the teats being the same, and then it's the mother who changes something, and then the milk qualities would change. So they just have basically different bottles as they grow, like for different ages.
And some mothers can have, for some species can have multiple pouch young at different stages as well. So, there could be like a pouch young on a smaller teat, and there could be like maybe a 6-month-old one on a larger teat, etc. So it's quite adaptable in that sense in terms of when they're breeding. I've actually got a video to show you if you want to have a look at the journey of the marsupial from, being born to the pouch in a kangaroo.
Yeah, let's do that.
Yeah, it's pretty amazing, really. How it works, your question, I mean, I have no idea. There must be some kind of signaling or scent. Maybe, the mother will kind of like lick, certain areas, and maybe will be attracted to that. But here is the video here. So you can see this is, that was the birth canal there. And now it's crawling up. Look at that.
What a little climber. It looks a little bit like a worm. I mean, of course, it looks kind of like a baby mouse or something. But because it has just been born, it looks a little bit, it's very tiny and squishy. But it knows where to go.
Yeah, it is. And this is now entering the pouch right now. You can see it's just crawling over. It's managed to make it up there. And this is just hanging off one of the teats. And once they attach, they're really firmly attached, particularly when they're at a younger age. They're almost fused to the teat.
Yeah. And actually in this video, I can see that it's the upper limbs that are more developed. It almost doesn't have the lower one. So that's what it uses to kind of climb up.
Yeah, no, definitely.
Yeah, but it doesn't have claws. It's amazing how it can actually attach and do all this climbing.
Yeah. No, it is pretty amazing. It's quite a journey, like if you're a small little Joey into an open world, it's trying to get to the safety of the pouch. It's quite amazing, yeah.
It is, and then it just hangs there. It's it's a very tough start in life, but they do it. And so this pouch is amazing because it has so many functions. And since this is the podcast of the Evolutionary Hologenomics, I'm going to make the bridge up to there because there's more to the pouch than just, and that would be enough, but then just being a safe space for the babies to live in and develop and feed. But actually there's a lot of microbes in there as well. So there is a pouch microbiome, and this is what you've been studying, right? So what can you tell us about that?
Yeah, no, definitely. So this is, let me just change to a slide. That's just some obligatory cute photos of Joeys that I thought I'd show people who are watching the video. It can be pretty cute.
Yes, and if people are just not watching the video and listening to audio, maybe you can Google some images of, is that a wallaby?
Yep, this is a wallaby.
I'm not Australian, so what's the difference between a kangaroo and a wallaby?
I think it's mostly taxonomic. So, people say it's this particular group. There's probably some characteristics that are a bit different, but for all intents and purposes, I think they're usually a bit smaller as well, wallabies. Kangaroos are typically the larger ones. So they can get up to, over 60 kilos or something, some of the big kangaroos in particular. But these wallabies here, these are the yellow-footed, so they only get up to about maybe 10 or 12 kilograms, a bit more nimble.
Yeah, so we're looking at, go on.
One of the right-handers.
Is that a wombat? But it looks yellow. So are they born, do they have different colors? Because the one we saw in the picture before, it was kind of gray. So do they change color as they develop, kind of like wild boars? They're very cute with spots and so on, and then they become dark? Or is it actually adults can look different colors as well?
So, I mean, from what I've seen, most wombats are pretty similar color if they're from the same population. You'll see maybe some differences between populations, but we're not really sure with this one here, actually. It could be that it's got quite light colored fur. And, maybe this is the pouch gunk that's kind of in there. It's kind of making it that color. Or it could just be a blonde wombat for some reason. Sometimes there's something random happens and there's just a little cute blonde wombat.
Well, for people who are Googling images, you can also Google blonde wombat and see what you get. I'm not responsible for the images you might find on the internet, just saying, disclaimer, but they are all very, very cute. Mm-hmm. So it must be very nice to do. I guess you did some field work as well for your research? Yeah.
So these are photos I've actually taken out on the field from work with some of these animals. So yeah, it is really great to do field work on these animals and see them out in the wild and how they interact with the environment. And then also just to, touch them and, see what they're about. It's pretty amazing. So
yes, that's your job. That's very, very nice. So back to the microbiome of the pouch. What can you tell us about that?
Yes. So, I feel like in the microbiome field now, we're expanding beyond just the gut. The gut has been a real big area of microbiome research and interest for many obvious reasons. But people are now moving more towards other parts, like what is the mouth, the oral microbiome, the skin, etc. And so I was thinking, well, why don't we look at the pouch? It's quite a distinctive feature in marsupials compared to placentals. But there actually hasn't been a huge amount of work done on pouch microbes. There has been a handful of studies, back in the 80s when they did, very old school techniques like culturing microbes. However, that has quite a few challenges. We try to grow microbes in the lab. A lot of them don't like to grow with the food that we give them, so it's very challenging. And so today, there hasn't been many studies using DNA sequencing techniques to look at the microbes and study them in the pouch. There has been one previously before the study. However, they didn't really account for a DNA contamination, which is a pretty big thing in the microbiome space that we're learning about. Do you know much about contamination, Hannah?
I do, but I would like you to explain to our listeners why this is important and why, especially in the wild where, you don't have a clean lab where you have only the species and whatnot and so on, and they're just everywhere and they contact with so many things. Why is it important to have that?
Essentially what the challenge is that the DNA sequencing techniques that we use, one of them in particular, this barcoding approach, is extremely sensitive, right? So we're doing multiple cycles of PCR, and this is when you essentially photocopy your piece of DNA. We can do multiple cycles of that, so we do multiple reactions. And so we can start from one molecule and end up with like billions or of molecules, essentially, because it's an exponential kind of reaction. And so what this means is that if you have a biological sample that doesn't have any microbial DNA in it, you can actually pick up DNA from the environment, if we're working out in the field or in the laboratory. So people have shown now that reagents that we use for these reactions can contain a bit of contaminating DNA. And so if you don't account for this, then you can get kind of a false positive signal, right? You might think, oh, look, we found a microbiome in this sample type, but actually, in fact, it could be just DNA contamination. And this has been a pretty big thing for the field, particularly for the placenta. So the human, placenta, there's, in 2014, there was a study that said, the placenta has a microbiome, which was met with a bit of skepticism, right? Because previously we thought that the placenta was a sterile organ, right? There's nothing living there because otherwise it'd be a danger to the fetus. And subsequently, since then, when people, scientists have kind of tried to re-verify this study, they found that there weren't any microbes at all. So the current evidence suggests that there probably isn't a placental microbiome, and that a lot of the results were from DNA contamination. So with this in mind, going into the pouch, which not many people have done before, we wanted to take a bit more of a rigorous approach and really try to control our study here.
Yeah, and it's just amazing. So you actually touched on something that I know a bit about, because my master's thesis a long time ago was actually comparing how to identify bacteria using traditional microbiology methods and then sequencing. But by sequencing, I mean, it was not NGS or even 16S. It was, a long time ago. So as you were saying, whenever you try to culture bacteria, so that's one way of trying to figure out what's there, right? So you take a swab or something, then you put it in different plates that contain basically different foods and try to grow them. And then you can also put them at different temperatures. Especially I was looking at soil and bacteria that live in nodes in plant roots. So they can withstand very hot and very cold temperatures. So we try to cultivate them at different temperatures, as well as different CO2 levels and so on. And obviously you're going to get only a very small percentage, because you're not recreating the conditions they like. And then it's even harder. Those that grow, then you can differentiate them by color, by the overall aspect of the colony. Some you can put like under a UV light. You can smell them. I don't know about smelling your samples, but some people do that. So there's all these things, but still, so not only do you only get a very small amount of the variety, but you also kind of almost sure, but sometimes it's hard to say. And you look at something and you say, oh, it can be this or it can be that, and we're not sure. So then things have evolved into molecular methods, and that's a little bit more accurate. And it can be, DNA can be picked up from a much smaller amount. And as you mentioned, then we can do PCR, which is basically many copies of the same thing until we can detect and see what's there. And then I was doing basically I picked some genes that I was interested in. So these were nitrogen fixing bacteria. So I chose some genes related to that. And then I kind of lined them up for the most species I could and designed primers for the common regions. So then I would amplify that and send it off for sequencing. But now at the center, especially, we all work with either 16S or 16S and mostly combined or alone with this next generation sequencing, which is basically you start out of nothing almost, and then you just basically sequence anything there is in there. And then you just get all the information of all the species that can be found there. So that's amazing. But going back to your point, it does amplify everything and it does detect everything. So if you sneeze on your samples, it's going to go in there. If you don't wear gloves, your hand microbiome is going to go in there. If a wombat has been into different holes and, exposed to, I don't know, pollen and all these things, all this is going to be detected. And then how do you separate between what's the microbiome in there that belongs there and does things and what's other stuff? So this is where the controls come about. So how do you collect and what kind of controls do you do to then separate this?
Yeah, so we wrote a paper about this. Some of the best practices for dealing with DNA contamination in microbiome studies. So if people are interested, I can try and provide a link at some point, or maybe we can upload that a bit later. But essentially, we have a number of different types of controls. So the negative controls are quite very, very important ones. So these are kind of samples that we collect out in the field. In this example, we'd basically have we were collecting these samples with swabs. So what we'd do is we'd basically just open up a swab sachet, expose it to the air for 5 to 10 seconds, just to kind of pick up anything that's in the air, and then just put that into a tube, and then just basically leave that until we did the DNA extractions. So essentially what we're looking at there is just basically, is there any DNA that's on the swab itself, from the manufacturing process? Could there be some DNA that was introduced in? But also, what kind of microbes are out in the air when we're taking the sample? So which we can then use later to kind of look at and get rid of that signal. We also have extraction blank controls. So when we're doing the DNA extraction, we'll basically have just a blank tube that we do that reaction on. And again, just kind of looking at microbes that are present in the reagents or in the laboratory environment, for things that I might introduce to the samples when I'm working with them. And then we also have PCR controls. So, these are basically blank reactions of the PCR reaction. So we can kind of look at what's potentially being introduced in the PCR reagents. And then we can ultimately take all these different negative controls, and then we can process them once we have all the data to kind of ask questions like, what microbes are more prevalent in the negative controls than they are in the biological samples, et cetera. And then we can use that to kind of identify and get rid of those contaminants.
Yeah.
So those are negative control types. There's also another type of control, which I'll explain maybe in a few more slides when we get onto the data, which is the kind of the positive control. And that deals with how many microbes do I actually have in a sample in a rough sense.
Yeah, so let's get into that. But and there's also another type of control. So for people who are not scientists and kind of wonder what the heck do we do and how do we get our data, we do think a lot about statistics and about experimental design and all kinds of controls to make sure that the data we get really represent what we're trying to study. So all of these that you mentioned, positive and negative, they're kind of biological controls, if you will. And then we also do other things called technical controls, which is to do with the techniques and the reagents that we do as well. So they're not so much to try and see if there's contamination, but to try and see if there's a problem along the line of the process. So then if we do all these sorts of controls, and then we can kind of subtract all this information, that is the extra stuff that is not related somehow to our experiment, then hopefully we get our precious data that are the aim of our study. But yeah, so let's move on and see some exciting results.
Yeah, cool. So I'll just briefly just mention, so this work, this is actually, I was supervising a veterinary master's, Cecilia Weiss, who is actually pictured here. And what's very interesting is she's actually from Copenhagen, which is pretty cool. And I'm going to be heading over to Copenhagen soon. So it's a small world in that sense, I guess. So yeah, maybe before we talk about that, do you want to know a bit about what a wombat is?
Of course I do. And I'm sure our listeners want to know as well.
Yeah, so wombats are these quite interesting animals. They're actually quite closely related to koalas compared to any other marsupial. So, which is quite interesting, right? You think of these, bears living in the trees. They're actually in the same family, which is called Vombatidae, as wombats are. And so wombats are these, stocky, muscular quadrupeds. They can range between, 20 to 35 kilograms. So they can be pretty heavy. And they're all herbivorous. So they'll eat grass and do a really good job of that in terms of their niche. A collection of wombats is called a wisdom. I thought that's a pretty cute little fact. The wisdom of wombats, pretty wise little animals. And they basically live in these burrows. This is one of the key adaptations to their environment, right? Particularly the hairy-nosed wombats, which is the ones that we're working on in this study. So they live in these burrow systems, and you can actually see them from space, which is quite amazing. This is kind of a picture taken from space. And you can see these little areas that are kind of like where the grass has been chewed away. These are the kind of the burrow entrances. And it's really cool as well. You can see these little kind of almost like networks between the little burrows. So they're obviously doing some very interesting social kind of networking in this particular example. We don't have a huge idea of how this all works yet, but I thought it was pretty cool. You can see them from space, which is pretty cool.
That's very wild. Do you know what is the scale of this picture? Because it is very obvious, very clear.
Yeah, I don't have the scale bar for this one, unfortunately, but it's pretty amazing that you can see, these animals have such an impact on the landscape that you can see them from up there. They also, because they have quite a big impact on the landscape, there have been a lot of conflict with farmers as well in the past, like they'll dig off, fences and stuff like that, and everything. But so the key reason why they have these burrows, particularly the southern hairy noses, is that they escape the temperature extremes, right? So as I mentioned earlier, we've had some pretty hot days here in Adelaide, 45 degrees Celsius. If you go a bit further away from Adelaide, a bit deeper into South Australia, it gets even hotter, the further you are away from the ocean. And so, it can get over 50 degrees Celsius over there, which is just like, how does anything survive out in that environment? And so the wombat's solution to that is that, well, I'm just going to dig a little hole. I've got a bit of air conditioning in there. It's kind of like very well regulated, the temperature. And so it can be 50 degrees Celsius during the middle of the day, but it'll be only, maybe 23 degrees underground, which is just like, it's a pretty good solution.
Yeah, and that is crazy, the temperatures. But I guess humans learned from animals, because I've watched some documentaries about Australia and like extreme living, and people do that. They basically make their own fancy burrows where they just hide from the sun.
Yeah, there's actually a little town, I think it's called Cooperative, which is in South Australia. And that's the one. They have these amazing, it's almost like an underground town, which is quite amazing. That could be us in 100 years if we don't get, climate change sorted. But
maybe we develop pouches as well and we all become wombats, who knows?
Yeah, so these are wombats essentially. There's a few other little interesting facts. Their teeth. They have this diet of grass essentially, and they can live to quite a great age, so up to 30 in captivity, maybe 15 or 20 in the wild. And so if you're eating grass, it's quite rough, right? Day in, day out, it wears down quite a lot. So an adaptation that wombats have developed is that they have teeth that basically constantly grow, so that when their diet is wearing out their teeth, it's just going to be replaced. And this is an example, I guess, of convergent evolution with rodents. Rodents have these incisors that keep growing because they're gnawing at things constantly. By convergent evolution, I mean these are two very different mammals or animals, have very different evolutionary histories, but they've kind of come and converged onto that same phenotype, which is pretty cool.
Yes, thank you, Professor Raphael. Here we do like to educate people, so thanks very much for that. Since you touched upon convergent evolution, can you tell us a little bit about divergent as well? Because then this is kind of like a little parenthesis textbook moment of the types of evolutions we have, which can be roughly divided into two, which is the convergent and divergent. So what would be divergent evolution?
Good question. I don't know.
So basically it's the opposite, right? So convergent is when two different things somehow develop similar things, either because they want to perform the same function or whatnot. And then divergent is when you start from one and then they separate into different things, but they started as some kind of a common.
So I guess the example for that would be marsupials in Australia. I think based on some fossil evidence or some other form of evidence that Australia was populated by one type of marsupial that was probably of a single form, and then that diverged off into all these crazy wacky shapes that we have now in Australia. It's a good one. Yeah, I could talk a bit about guts, but I've got a lot of wombat facts, but again, I might be diverging a bit too much from the story here, but maybe we could just push on. What do you think, Anna?
I mean, that was moving on. Let's talk about guts now. But maybe just a little bit, because but it's very refreshing to talk about the pouch microbiome, because usually and it's not a bad thing, but most research that I'm involved with and that we do at the center somehow is gut related, although we do have researchers, hopefully in future episodes, that study microbiomes of other things, other places. But yeah, so we can go into the gut a little bit and then we go back to the microbiome of the pouch.
Yeah, no, sorry, I just thought it was very interesting. So they're actually pretty amazing herbivores. They don't need to drink water, which makes sense in their environment. In South Australia, where it's so hot, there's not a huge amount of open water sources. So they actually drive all of it from their diet, from their grasses, and they're super efficient. I think people did some physiological research on them in the 90s, and they estimated that they get, in terms of how they actually get that energy from their diet, it's this fermentation of that fiber from the microbes. And they estimated that over 60% of the energy intake from the wombat was from the microbes that are living in the gut. And wombats have pretty enormous guts. So I think it's about 11 or 12 times their body length. So wombats are about 1 meter long. And so that's about a 12-meter gastrointestinal tract, which is just enormous.
Yeah, but if it needs to do all that, and it's quite amazing that they don't need to drink water. So definitely they need to have a very healthy microbiome, not only to digest everything, but I guess to digest it properly in a way that, because obviously plants do contain water, but you wouldn't drink some grass. So to be able to extract water, there is enough for survival. That's quite a feat.
Yeah, no, it's pretty amazing animals in that context. And so I'll just give a little backdrop, because it's a very picturesque area. This is just the kind of area that we did the field work. So it's about an hour and a half drive from Adelaide, a place called Swan Reach. And it's on the Murray River, which is one of the largest rivers, I think maybe the largest river in Australia. It's pretty remarkable feature of the landscape. If you think of Australia as quite deserty in some areas, particularly in the central part of Australia, the Murray is like an oasis. It's like a river oasis on the landscape. This is a photo that I took on the river. We did a tour on the river on the boat, which is really fun. And you can see that these cliffs are just stunning. This is actually kind of limestone slabs, essentially. And so geologically, I'm not a geologist, but people have told me that essentially this cliff was essentially underwater, under the ocean, millions of years ago. And these are actually limestone sheets, which are basically, the sedimentary layers of the seafloor, which have basically fossilized all that marine life over time and put layers upon this. And it's pretty amazing that, it's been buried for millions of years, then it comes back up, and that forms a new, important aspect of current living life. So we have birds living up on these shelves. We obviously have the water that's going through it. It's pretty amazing. And this is actually a little fragment you can see from that limestone cliff. So you can actually see the marine fossils fossilized, some macro kind of shells. And then if you used a microscope, you could see all these little tiny, single-celled or multi-celled microscopic animals, which is pretty amazing.
Yeah, that is very cool. And so I did study marine biology. So we did a lot of field trips to look at mountains, and some are exposed, and you can see the different colors and the different heights of the layers. And so that kind of tells you what kind of material was being deposited, and also how long it's been deposited for. So if it's a short or a tall layer, then that obviously occurred in a short period or a long period. And then if it's kind of like argile, then it was the level of the sea was deeper because the particles were smaller, so they needed longer to deposit. And then if it's more organic, then it has different colors. So yeah, looking at that very yellow, you can tell it's organic marine. And also it doesn't look super compact like granite and so on. So you can see that you can almost easily kind of, destroy it. Let's not destroy nature, but it's not as solid as granite. And it's amazing, as you said, that it was once in the bottom of the ocean, and it was the habitat for these species. And now it's up, and it can be the habitat for very different species of birds and very cute wombats.
Yeah, no, exactly. And so that's a very good little segue into this. So some of these wombats actually use these cliffs, these limestone cliffs, as little burrows essentially. For a wombat, it's really great because these are these big slabs of limestone that have already been formed, and the wombat just has to do a little bit of burrowing for some of the soil, and then as a result, he or she can get underneath and have a really nice habitat. So this is a wombat we took a photo of near one of these cliffs, and it had a nice little river view burrow essentially, which is pretty amazing.
Location, they know it.
That's the one. And so we obviously sampled the pouch in the study. We're interested in the pouch, but it's very hard to get a pouch sample. You don't just go up to a wombat and ask them, "Hey, can I take a swab of your pouch?"
You don't?
No. If they see you, they're running to their burrow.
Hey, mate, here's a swab. No.
And so when I started thinking about this, I thought, how the hell am I going to get these samples? And the answer is you actually get in touch with someone who studied wombats for decades, an expert on wombat ecology and biology. And so that's Dr. David Taggart, who's a good friend and colleague of mine, who's helped with these studies enormously. If it wasn't for him, we wouldn't have been able to do the study essentially. But over the decades, he's come up with a way to catch wombats. And it's one of these interesting things. It's like the evolution of catching wombats. He's telling me stories about, 20 years ago, they started very suboptimal way of catching them, but they've, through, trial and error, developed a really amazing system. So this is actually a special wombat catching ute. It's got, special modifications that make it very good. Like it's a flat tray. You can kind of get off and on it very easily. there's basically these little holders so that when you're on the tray of the ute, you can kind of hold on and be comfortable. There's also even like a little red light. So wombats are very sensitive to noise when you're out at nighttime. And just like knocking on a car can kind of set them and basically make them run away. And so they have this little light switch here that basically when someone's on the rear looking at a wombat, they've spotlighted one, they'll basically press a button and they'll shine a red light into the side mirror of the driver. And that's the signal for the driver to stop driving and slow down. So little adaptations over the years, as well as with the weaponry for catching wombats, which are these giant steel poles, which are about 2 meters long.
Let's just clarify that it's friendly weaponry for
for
the sensitive listeners.
No, it definitely is. And so how we catch them then is we go out at nighttime. They're mostly active at nighttime because, as you can imagine, during the daytime, it's not very good to be outside in summer. And in general, they're not going to lose as much energy if they're out. And then I think, maybe historically, they don't want to be caught by predators as well during the daytime. So they come out at nighttime. And so we'll go out in the middle of the night.
Ask you, because I didn't know this word before. I don't know if it's an Australian word or just my lack of knowledge in specialized vehicles. But what did you call this? It looks like a van. Basically, it has like the front part of like a car. And then there's the back that is just with wheels and open. And then it has all this specialized stuff in there in the red light. But I had not heard this word before. I wonder if it's Australian.
It's called a ute. I think maybe it's like a pickup truck, the American version or something.
Maybe. Okay. Yes. Like an open back pickup truck.
Yeah, like an open pack. Yeah. Pretty handy little thing, obviously you can't take as many passengers, or you can't drive with many passengers on the back because, it's very dangerous, but very handy for
health and safety. Yes. And so this tool that is used to catch them, does it have a net? So it basically has this handle, and then it's like a square frame, and then it has a net in it.
Yep. So it's got a very strong net there because their claws are very strong as well. Really, really, really intensely strong animals. And so we have this little net. So we'll basically go out and net them out in the fields.
Yeah, they do look very muscular.
Yeah, no, they are. And they're very fast as well. So we'll go out in the middle of the night because that's when they're kind of most active. And we'll basically spotlight them. If we see one, we'll basically hop off the ute and go and chase them with a net. And, there's mixed success with that strategy. They're obviously very keyed on to us. They don't want to be caught. And they're also very fast. I think they can, apparently run up to speeds of 40 kilometers an hour. So we're talking super fast little animals, They're not going to, win an ultra marathon, but they can sprint. So it's a real challenge.
That is very fast.
Yeah. So once we've caught the animals, we'll take them back to a home base essentially. And then what we'll do is we'll anesthetize them because obviously we can't handle them when they're, conscious. They're just so strong. The 35 kilos of almost pure muscle. You can't handle that. And obviously for the animal itself, you don't want to stress it by doing that. So
yeah, I would imagine they will be very stressed. So that's the best for the animal as well.
Although very interestingly, when we net them, what we do is we transfer them to a big sack. And once they're in the sack, they seem to be very relaxed. They'll actually, fall asleep in the sack. I don't know if this is some weird, hark back for memories from in the pouch, like a nice little enclosed area, but I found that very interesting. But we'll take them back to the work base. And then we'll don our protective gear when we're taking our samples. So we're wearing gloves and masks here. Again, as you mentioned before, to kind of prevent us from putting our own skin microbes or mouth microbes into the samples. And we're using pouch swabs here for these particular samples. And so we collected a range of different samples from wombats because no one's actually looked at wombat microbes before, really. So it's very new ground. So we wanted to really collect as many samples as we could. And in the context of the pouch, right, we wanted to know, which sample type is more indicative of the pouch. Are there microbes from the gut in the pouch? Is a question, etc.
Yeah, and just so because some people are listening and not just watching, in case you wonder what a swab looks like, it's 2021. So, the COVID test, it really looks like that. Just to, for the people who are not looking at the video, that is exactly what the swab looks like.
Yeah, exactly. And I should also note, once we've taken our samples and people have taken their measurements as well, because TAGS is also studying these animals through time, seeing how they're kind of adapting to the climate and different, drought years, etc. He's taking, multiple measurements of the body as well. Once we've taken those measurements, we'll drop them back off at the warren that we quartered at, because these wombats, they basically don't spend a lot of time out, outside of their warren or like off their warren. So we catch them in very close proximity. When we catch them, we'll basically log in the GPS and say, okay, this wombat lives at this postal code. And so when we finish, we can post them back into the little burrows.
Home delivery. And are there any studies about how far from home they kind of forage and roam at night, or not really?
So yeah, I think TAGS has actually done a bit of work on this. So radio collaring animals and seeing how far they will move. And I think they estimated it's about 3 or 4 hectares. So It's not a huge distance really. Because if you're a wombat, why do you need to move around a lot? You've got your grass at your doorstep, you've got your comfy little burrow that's safe. I think mainly it's, potentially when it's breeding season, that's when they'll start to move around a bit more, Yeah.
And do they tend to live in the same burrow forever, or they, from time to time, they move either because the burrow gets old or because there are resources or something?
Yeah, it's a good question. I think we know now that it seems that the mothers will basically donate their burrows to the young, which is quite a nice little act, it obviously helps the next generation. I wish my mum gave me a house, but I'm gonna have to buy it one day if I ever do. But so that seems to be a factor there. But this is pretty amazing because I think some of these warrens, these burrows. So warrens are basically like a collection of burrows. So basically you have like multiple entrances or exits to a particular burrow. Some of these could be quite old, maybe centuries old. And so some of them have kind of almost been passed through families almost of wombats, which is kind of interesting to think about. Because as you can imagine, it's very energy intensive to create a new burrow, right? We're talking piling out tons and tons of soil and quite, pretty harsh minerals and such.
Yeah, it's hard work. So they have so they use their front paws, I'm guessing, and they have very long nails. Are those nails, or are there some kind of specialized structure? They're still nails.
Just nails. Yeah, just nails. Yeah, so very quite sharp and quite strong nails. Really good for scooping that dirt. Yeah. Cool. So once we've basically collected our samples now, we've dropped them off at home. And so then what we did is we took these samples to the lab and did our DNA extractions, our molecular biology work to basically amplify that 16S gene that you mentioned before, which is this little kind of barcode for microbes. We sequence that DNA, and then we can basically get that data back and start analyzing it and making sense of these different data. So one of the first steps that we have, like the first kind of analysis that we wanted to ask, this relates again to the contamination that we were talking about before, was how much DNA is actually, or microbial DNA is actually in these samples, right? Because as I mentioned before, like one of the issues, if you have a biological sample with no microbial DNA, that's very prone to contamination, right? Because basically that DNA can't compete with, it doesn't have to compete with anything at all. It basically just gets amplified. Whereas if you have a sample with a lot of microbial DNA, so say like a fecal sample, any kind of contaminant DNA doesn't really have a major impact if it's at a low baseline, low level of contamination, which is what we normally find in the laboratory, if you're doing lab work correctly, I should add. So this is the first kind of question we want to ask, how much DNA is in the different sample types that we collected? And so one way we do this is through a quantitative PCR, where we basically do the PCR cycle reaction, but what we also have in that reaction is like a fluorescent dye. And that dye, in this particular example, will basically bind to that DNA. And what ends up happening is like the more DNA you have, the more fluorescent signal you have in your tube. And so you can say like, okay, this sample has a lot more fluorescence, so therefore it has a lot more microbial DNA than another sample. If that makes sense.
Yeah. So just to clarify a little bit, sorry, just try to keep everyone in the loop. So this qPCR, as you were saying, is to detect the amounts. And as we explained before, so PCR is basically making many copies of the same thing until we can detect, because if we just take a sample, it has DNA there, but it's such little amount that we couldn't actually just detect it. And then, of course, if we're going to make many copies of all the samples in the same way, then, of course, if the starting sample had more, then you'll end up with more in the end. And if the starting sample had less, then you end up with less in the end. And so this is then how we can assess which one has more or less in the end, which reflects if it had more or less to start with. So this is kind of how we quantify things. And then you have very interesting circles here. So we're looking at a graph about the amount of the DNA in the different samples, and then there's different colors of circles, and they seem to be for the same sample very specific. They're all clustered together, so they're not mixed all over the place. So this is actually a really nice graph. Can you tell us a bit more about it?
Yeah, great explanation as well on the qPCR. So here we have on the x-axis the different sample types, and as you mentioned, they're colored based on what sample type they come from. And then on the y-axis here we have the amount of microbial DNA, or what we call the CT value. And basically this means it's actually kind of counterintuitive. So the higher the CT value, actually the less DNA you have, and so the lower the CT, the higher amount of DNA you have. And so the general trends here are that fecal samples, as we expect, have a lot of DNA. So they are the samples with the lower CT values. And then the negative controls, which basically we didn't put any DNA into, or we didn't on purpose put any DNA into, they have the lowest amount of DNA. And the samples that we're interested in here, this is the pouch, two different types of pouch samples that we have. They have kind of intermediate amounts of DNA. They're actually quite a bit of DNA compared to the negative controls, right? And so this analysis is very important if you're studying a new sample type, something that people haven't studied before, and you don't really know yet, is there DNA in the sample yet? Are there microbes here? Because, I feel like we always assume that there are microbes everywhere, which is, I think based on a lot of good evidence, right? Microbes kind of rule the world. But at the same time, I don't think, there are scenarios where there aren't microbes, around in a particular area. So this kind of analysis will help you get it into that particular question.
Yeah, I just had this random thought. Maybe I should have a little hat that I put on and off whenever I'm explaining something, because I just had this urge of explaining a bit further when you were talking about CTs and whatnot and why it's counterintuitive. So basically, we're looking at this graph, and as you mentioned, the higher the number, actually the less DNA there is. And this is because basically what we're doing is making multiple photocopies of something until it reaches a limit when we can detect it. And so CT is this cycle threshold. So how many cycles do we need to do? And so this is why the more cycles you need, then it means the less DNA there was there to start with, because you need to photocopy it so many more times to actually see something. Whereas if you need to photocopy it just a few times, it means you had a lot of DNA there to start with. So this is kind of how we can detect. And just one last note. So I don't know if you touched upon that for the people who are just listening and not watching, the different samples that we have here and what is the one with highest and lowest amount. Can you go through that?
Yeah. So the sample with the highest amount of DNA is the fecal samples, which we kind of expect based on our previous knowledge on other animals. And then the samples with the lowest amounts of DNA or the highest CT values are the negative controls. So these are the sampling controls and then extraction plan controls that I mentioned before.
Yeah, and you also sampled milk, joey, pouch lactating, and pouch subadult, which is different stages. And I will ask you a little bit more about that very shortly, because I know you're dying to tell us more about that, because it's very exciting. But I just have a question I saw here. So milk, I guess you take a liquid sample, but joey, what is this joey? Do you swab the baby or?
Yeah, essentially just swab the joey, like the skin of the joey, and basically look at that.
Okay.
Yeah. And so I guess the take home from this is that we actually have microbial DNA in these pouch samples that I've highlighted here in this box. And that there's actually quite a lot more DNA in these samples than there are in the negative controls. So we can conclude from that we haven't contaminated biological samples, and that we actually do have microbial DNA in these samples, which is, I think, a very important and underappreciated aspect of microbiome research. So
yeah, definitely. So get things straight. First things first, experimental design is very important. So before you launch into doing all these, because some people do have the money, so they're like, oh, let's get samples randomly and not wear gloves and just do next generation sequencing and see what's there. And then there's many problems with those. So a step-by-step approach where you actually, first compare the different bits, and already here at the different locations, and already here you see differences in amounts. So you don't know what's there yet, but you already have this base data where you know that there's a very big difference between your negative controls and your actual samples, which is good. And then you also see that there's different amounts of microbial DNA. And how do we know this is microbial? Obvious to you, but please do tell what that 16S there means and why this is microbial and not kangaroo or wombat.
Yeah, no, good point. So the 16S is for the 16S ribosomal RNA gene. So this is like basically a housekeeping gene. It's a very important function. It goes part of the ribosome, which is the part that basically creates the protein. So in other words, it's an extremely important function for any living cell. And as a result, it's basically in, all animals or microbes, etc. But there are distinct differences between microbes and animals and their ribosomes. And so we leverage this in this example by targeting just the 16S gene itself, which is different to the animal one.
So exciting news for this first step. And then you can move on to figure out the differences and finally what's there.
Yep, no, exactly. Cool. Okay, so the next question we kind of ask as a basic kind of microbial ecology question is like, what kind of diversity do we have there? So how many different types of microbe do we have in the different samples? And so here's a figure. It's a box, whisker plot figure with also the individual data points. And so again, on the x-axis, you have the different sample types. We obviously didn't test all the sample types because we were mainly interested in the pouch in the previous figure. And we didn't really need to know, like, are there microbes in the mouth, for example? That's, I think, probably fairly obvious. And on the y-axis here, we have feature richness. So this is just basically the number of different kind of sequence types that we had of microbes. Now, this doesn't necessarily mean, different types of species. There's a bit of a debate at the moment about, how specific this is. We know, for example, that microbes can have multiple copies of the 16S gene in their genome, which means in some situations you can have, 7 maybe different copies of the 16S gene. And so when you amplify that microbe, you might have 7 different sequence types. But then that's a little, detail anyway, maybe a bit more of a unnecessary detail for now. But essentially here we're looking at the richness. So how many different types of microbe are in the different sample types? And we can see, skin has quite a large diversity, which is what we expect, right? Because these wombats are quite low units. They're in contact with the soil a lot. They're probably picking up a lot of microbes from their environment. And we actually sampled the skin from near the pouch, so about 5 centimeters from the pouch as well, just to see, like, are there any microbes from the skin going into the pouch, etc. That was kind of like a little control in that aspect. Then we also have the pouches of the subadults. and the fecal samples, which we know fecal samples are quite diverse anyway. So you had a question before about, what are the different kind of pouch samples we have? And so I'll explain that a bit now. So as you'd imagine, these animals go into reproduction different times. And so when we actually sample these animals, we caught a range of different animals at different reproductive stages. And so we have animals that aren't reproductively active. And so, basically they're not actively breeding. We have subadults. So these are, basically youngsters. And this is what a subadult pouch looks like. It's almost like a belly button, you can see here. Very, shallow, very dry. And this is a pouch from an adult, which is actually breeding. It actually has a pouch young in it right now. And as you can see, it's almost like there's some kind of like secretion, some fluid out there. And it's very deep, the pouch. Like the whole kind of physiology of the pouch changes through the reproductive cycle, which is pretty cool to see. And so the key finding here is that the pouch from the subadult is quite diverse. It's almost as diverse as the skin. So we kind of say like, there's not a huge difference in terms of diversity between the skin and the subadult pouch. But when the wombat is actually breeding, that's when the diversity plummets. So, we go from having, hundreds of these sequence variants, these different microbe types to, maybe 20, which is quite drastic.
And do you know why that is? I don't know if you were about to tell us that or not, because and I think with a lot of microbiome studies now, we're actually finding that because people have this idea somehow that diversity is important, and the more diversity, the better. And now there's a lot of studies that suggest that for specific functions or periods in life or whatever, actually lower diversity is better.
Yeah, I mean, I guess this is the thing, right? We always like to say this way is this way and, nothing else, but biology is very complicated, right? And as you, alluded to, like diversity can be very good in some contexts. Having more diversity can be great. It can lead to a more resilient ecosystem in some contexts, but in some situations, it can actually be better to have, low diversity. And so what we think is happening here, essentially, is that the host is, the wombats are filtering out what microbes can live in the pouch. That's our best guess for now. And the reason is, right, like these joeys are born very young, right? They're born between 3 to 6 weeks old. They actually don't have like a functioning immune system, right, at that time. And so when they made that journey, as we saw earlier, up into the pouch, they're crawling out into this microbially rich world, right? And they could be prey to some, nasty bugs that come along and infect them. And so what we think is happening is the mother is, when she's reproductively active, she's somehow filtering out these microbes, or, choosing maybe the right ones. That's a bit speculative now. That's something we're really interested in looking at into the future. But basically choosing which microbes can live, or just getting rid of, a whole range of different microbes to kind of make it like a sterile site for the joey.
That is so interesting, this dynamic, because people, well, again, this was probably a long time ago, this thought that things were separate and there were different boxes, and now it's the host, or the mother is not talking to the microbiome in words or kangaroo sounds, whatever they sound like, but it's all, I guess, chemical cues that the pouch environment changes in a way that then all these molecules and all this crosstalk with the microbiome then actually changes the composition of the microbiome, and for the specific function then of having the little one growing there healthy and free of disease, and probably also these bacteria might help with the development. So not only keeping away the bad things, but keeping in the good things. That's quite amazing.
Yeah, definitely. And so, we still don't really understand how this is happening, right? There's been some studies done on other marsupials. I think in koalas, actually, they isolated some antimicrobial peptides that it seems the mother produces. However, it's still pretty preliminary work, but there do seem to be some, potential antimicrobial peptides that might kill off certain types of microbes. So that's like a form of filtering out the microbes, right, from the mother. But then there could also be, maybe there are some beneficial microbes living in the pouch that when the conditions are right, as you mentioned, they kind of bloom and the host, by having them there, it's beneficial because maybe these microbes might help them fight off other bad bacteria. And maybe they even, perform important developmental functions with the host as well, right? Because these joeys are exposed to them at such an early age. Maybe they're training their immune system a bit in the pouch.
That's very cool. And so this is the work you've been doing so far.
Yep.
And now,
slide. Sorry.
Oh, you have more images. Let's go over that then.
Okay.
So, that's very interesting and very colorful. But obviously for the people who are just listening, there's no color. So if you could explain, and also even the people watching, they look pretty, but you don't necessarily know
a lot of
what's in there.
Yeah. So I'll just explain, because this is kind of coming at the question of, okay, so we've looked at the diversity, we've seen that that's shifted, that's dropped, right? When wombats are reproductively active. But then the question is, okay, what type of microbes are in the pouch, right? Is it just a random jumble of whatever managed to survive, like a lottery? Or is it quite a specific type of, set of microbes? And the answer is, based on the samples we had, about 16 or so reproductively active wombats, the microbes were all of about 4 or 5 different kind of microbial genera. So quite specific. There wasn't a huge amount of variation in that. They were very, almost like a core microbiome in the pouch. And so this figure here is just this taxonomic bar plot, is what we call them. This is basically, we have the different colors which represent the different types of microbes. And basically their sizes, represent how abundant they are, or, what their frequency is in that community. And so here we have more reproductively active wombats from the right-hand side less reproductively active wombats on the right-hand side, going all the way to more reproductively active. So we had like a spectrum, right? So we have subadults here, and skin, and then we see that diversity dropping. It's another kind of way of visualizing that drop of diversity.
Yeah. And so now finally we have, it's kind of like a nice little summary of, you started with some questions, then you went to do your controls and just to check it. Are there any bacteria here at all? Are they different in terms of amount, and what's the most interesting thing to study? And then finally you sequence them, and now you can actually see what species of bacteria are there. And also the distribution, if they are, more or less if they are reproductively active or not. And what we can see here for the people just listening is that in the less active, as we talked before, there is almost like this rainbow, a whole spectrum of so many different colors, which basically means so many different species. It's such a big variety. And then as they become reproductively active, we have only, a few different colors. And then that's the microbiome that was selected that is good for the baby. And so it's a very self-explanatory picture in terms of just colors that tell you, the number of colors tell you just how many. But then, of course, you went into the actual sequence and which species are there. Do you know, are there any new different species, or it's just like some disappear and then some prevail, but no new ones appear? Or is it actually completely different in some cases?
So actually what we think is that these microbes here that seem to dominate, they seem to also be a normal part of the skin or, the subadult pouch flora. But it seems that the conditions are just right for them to move. And I guess that kind of maybe makes sense, right, from if we think about potentially how these microbes are maybe passed on through generation. If this is a thing, we still haven't demonstrated this, right? This is still tentative, right? We need to do experiments to prove this. Obviously, the joey is born in that pouch. It's exposed to these microbes. They become part of the joey's skin microbiome, maybe. And obviously, the pouch, when they're young, is, like skin, for more or less. Maybe these microbes then live in that kind of pouch fold, the skin, as it's growing up. When that wombat then becomes reproductively active, that's maybe when these microbes kind of sprout up and, really dominate that particular community. So there is a potential idea for some form of vertical transmission, right? Like passing on the microbes through generations. And that actually leads to the next point is we asked the question, okay, well, what are these microbes that we found? We found that they're quite different to ones in the databases. So there are some differences, to the modern, reference sequences we have in the databases. But what was quite interesting is like 3 of these 5 kind of dominant ones in the reproductively active pouches, their best hits were to other pouch microbes that have been isolated from wallabies. So there was actually a study over 10 years ago now, where they did some kind of culture-based one, I think, technique that you were mentioning before. And basically, although it actually wasn't culturing, it was the old school, like Sanger sequencing of the 16S gene. So before we had NGS, which makes it a lot easier when you actually had to grow microbes and, insert that DNA into them. But they basically looked at the pouch and they basically sequenced some of the 16S gene. And these taxa here, the best kind of hits we have from the wombat pouch study were to these wallaby pouch microbes, which is kind of interesting.
Yeah, so it seems like these, is it species genera? No, these are genera.
Genera, yeah.
Yeah, so and do we know what they do, or we just know for now that they're good for the pouch when you have babies, and it's actually very interestingly present in more than one species, so it seems to be a marsupial thing.
Yeah, maybe so that our interpretation is that maybe there is some like co-evolution of these microbes with marsupials throughout the marsupial tree. However, we can't say that for sure. This is just very speculative at this stage. That's why what I'm really interested in looking at in the future is maybe like sampling pouches from other species and using the same methods, and then looking at, can we see this similar trend as well? In terms of what they're doing, as you asked, it's very hard to tell. We can say like, what does a Corynebacterium generally do? But they can be quite diverse, right? Like taxonomy is in general a pretty good indicator of what microbes can do, but there are always exceptions. And when we look at these microbes that are part of marsupials, which have evolved separately from us 170 million years, like there's only so far you can push that interpretation. And so as a result, we don't have an idea yet, but that's something that we're working on at the moment too.
Yeah. Maybe you can get some RNA or something, and then RNA. You can check, because it is true that even one within the same genus, there can be different functions, and also the same one in different environmental conditions, then they can be performing different functions. So here you studied DNA only, which means you can find out how much there is and what they're doing. Then there's other things like RNA, which then you can actually convert to DNA and amplify, because if you look then at proteins, which is the actual thing. So you have DNA, which is like the instructions. Then you have RNA, which is kind of the middleman, the little message. And then finally you have proteins, which is usually the thing that goes and do something. And so that can be internally or secreted, and it can function between species and so on. But protein, so far, we don't have a way of making more of it. So I guess it would be hard. We have in vitro, but if you just collect the protein itself, then you can't photocopy it as easily as we can with the DNA or RNA if we then convert it into DNA. But it would be interesting then to know the function, but one step at a time. And this is a lot of work. So for people who might not even realize nowadays with such an amount of data, because you explained it and you made it look so simple, but this kind of work actually involves not only biology, molecular biology, some field work in the beginning, but actually a lot of bioinformatics, which is something you didn't mention here. But this is how you then in the end get, you collect all this massive amount of data, and then to come up with these very nice graphs, you don't just snap your fingers. There's a lot of computer work involved here, just for people to have an idea.
Yeah. Oh, definitely. Yeah. Computer science meets biology. Bioinformatics is a really big part of biology currently, as you mentioned, because we have so much DNA sequencing data and we need to try to make sense of that. And really, the fundamental data we work with is just A, C, T's and G's, so 4 characters. And yet we can learn such amazing stuff about life with it, which is quite remarkable.
Yeah. And you just mentioned one thing that I kind of forgot when I was explaining about these traditional methods, and what I was doing before that. You try to culture these bacteria and then you make PCR of these genes with these common primers, that then you get this. And then I said you sequence it, but you mentioned something very important. So when we talk about PCR, we have the DNA and we make many copies of it. And then you mentioned briefly about having to grow the bacteria. So this is another thing. You don't, in these methods I was using before, you don't just take that PCR product. You actually, because then it's a mixture of the same molecule from many species, and you want to have that separated. So the Sanger sequencing. So then you have to take that and clone it into a vector, which is like a little plasmid round thing. Then you have to put that into bacteria. Then you have to grow them. Then you have to select different colonies that will have the different copies, and you want them to be each one an individual colony, which means each colony will have its own copy of that molecule for different bacterial species. Then I'm already tired. It takes so long. So it's so nice now to have next generation sequencing in all this, because this was so much work in the lab just to prepare the stuff. So then you get that plasmid from the different bacteria. You have to check what's what. And then finally you send off for sequencing. So it is a very long process. And then you're targeting only for what you think is there, or for the, it's only as good as the primers you designed. So potentially you're already missing out on a lot of things. So things have moved on a lot.
I'm glad they have.
Yes, and bioinformatics has so that's computer power, brain power, and bringing a lot of things from computer science and math and physics and all these models that biologists didn't used to do in the past, because we didn't need it, because we didn't have this crazy amount of data that we have nowadays.
Yeah, exactly.
So that's very good. So you've been doing this in the past 2 years, and now you're going to be doing more of it. I guess you've recently joined us here at the Center for Evolutionary Hologenomics, and we actually, the first episode we did for this podcast was with Aoife Leonard, and she works in the Earth Hologenome Initiative. And I understand that your next step is it within that project or a sister project of that?
Yeah, so it's basically a project nestled under the EHI. And so I have to thank Aoife, obviously, for doing a lot of the real hard work of setting up all the kind of, her and Anton and everyone else involved, setting up a huge amount of that work of getting the EHI framework started so that we can start creating these other projects.
And so it's a project of its own. Oh, you have
I have a little slide for you to show you.
Now we have Convergent Evolution, which people know all about it because you've already explained it.
I've already talked about it. But I'll give you another few quick little examples, which I think are pretty cool. So on the left here, we have different placental mammals. So from this little group here that we talked about, and on the right, we have different marsupials. So these are some really cool examples of convergent evolution that visually you can see there's a huge difference. And these little stars here that I've pointed next to them, I basically found which kind of mammalian family or order they come from. So you can see that like these flying squirrels and these gliders, these marsupial gliders, they look very similar, right? Like they've cracked onto this particular body form, but they actually are from completely different parts of the tree, the mammalian tree. And likewise for the moles here, This is the marsupial golden mole, which is pretty amazing, very rare as well. And then, do you know what this one here is, Anna?
Is it? Oh, it's extinct now, isn't it?
Yeah.
Is it from Tasmania?
Yeah, so this is the Tasmanian tiger. Although they used to be on the mainland a long time ago as well of Australia, but they're called the Tasmanian tiger because that was when, the last population or living population before they went extinct was located in Tasmania. As you can see, they kind of look like a dog. That's the kind of shape that they have. Obviously, dogs have many different shapes, but that's mostly due to us now.
And I'm still fascinated by this mole because I know the other one, I would say, is the regular mole. Probably I don't know about Denmark, but in Portugal, we have those in the backyard, or very similar looking ones. But this golden one, you say it's a marsupial, so the other one is not. But is it eating a lizard? And it has gigantic claws. I'd never seen this before. So what's the name of this so that people can Google it and find an image if you're just listening, because it's a thing.
It's pretty amazing.
Need to see it. What is this?
So I think if you Google marsupial mole,
yes,
you'll find it. But it's quite an amazing animal.
So it's a carnivore. I'm guessing it's not hugging the lizard; it's eating it.
No. Yeah. And
it's got these almost like a lobster kind of claws. It's a very interesting looking fellow for sure.
Yeah, no, definitely. Yeah. So these are examples of convergent evolution, right, between marsupials and placentals, some real kind of eye-opening examples that we can look at. And so the project that I'll be working on is one called Convergence Hologenomics. And this is where we'll basically be taking these kind of comparisons between placentals and marsupials from different ecological niches, right? So we have examples of carnivores. We've got like the domestic cat. We've got quolls, which are kind of marsupial cat, but these are these animals that have filled up the cat niche in Australia. And then we also have South American marsupials that are carnivores as well that we'll also compare to. We've got herbivores. So from different types, we have the hindgut fermenting. So that's like a wombat here. And then we also have foregut fermenting. So there's different ways you can kind of eat plants in the animal kingdom. And kangaroos are one of these kind of foregut fermenting marsupials. Then we've also got some small generalists here as well. So these are these kind of like mice and things that look like a mice. This is a little Antechinus, which is a marsupial kind of mouse, essentially. Really cute little fella. And then we also have mimicophages, which are anteaters. Fancy word of saying anteaters. And so there's actually a whole range of different anteaters, which have kind of convergently evolved in the placental tree, which I won't go into details about. It's quite interesting. But we also have marsupials, right? We have the numbat here, which is a really cute little anteater from Australia, marsupial. And we also have the echidna, which is one of the monotremes that we talked about earlier. This is actually another type of monotreme out there, and it's also found in Australia. And so the goal for this project is to basically sequence the host genetics and the gut microbiome from these different animals, more than just that I've showed here, and basically ask the question, like, what do we see that's different or maybe the same between these animals that have converged on this particular ecological niche? And so I think the reason this is a really powerful approach is, if we were to just take one of these animals and ask the question, what animal genes are important for its lifestyle? Or what microbes or microbial genes are important for its lifestyle? It's very difficult to get at that question, right? If you've only got a single species, there's a lot of genes that you'll see, but you don't know if they're important or not. But hopefully by, basically doing multiple comparisons between very similar animals, you might say, okay, look, we found this gene here that pops up, or, it's very highly, in abundance, or maybe it's very low, or these are particular microbes or microbial genes that also pop up more often than not. And so we're hoping we can help identify and figure out how these animals work a bit better by doing this approach.
That is very cool. And we kind of come full circle. We started a little bit talking about convergent evolution, and now here we are again. So it is very, very interesting that you're going to look at host and microbiome of animals that look similar in a way or have similar functions, but then they are totally different, and some are placentals and some are the marsupial sort of counterpart in a way. So
the alternate universe animal or mammal.
Yes. And so, yes, not everything that looks one way really is that. So we really need to look deeper, just because two things that look very similar, they might actually be very, very different on the inside.
Yeah, there's many ways you can get to that particular, goal that you want, have, right? Like, but, sometimes you can have, sometimes in biology, there are really good examples of convergent evolution where we've almost seen the same changes on the same gene, or the same, different type of function, which is really cool. So yeah, I think there's a lot of really interesting stuff to learn here about evolution and how these animals work. Yeah,
and if something works well, then you don't need to reinvent the wheel. You just make the same wheel in a different branch, sort of. And I was going to say, just because it looks the same, it's not the same, but actually, or is it in terms of microbiome? Because this is kind of your question. They look different, but since they converged for specific reasons, usually for a function or something that is an advantage, then it would be interesting. It's kind of like, it doesn't matter where you came from. If you do this specific thing that works, similarly to actually having the same genes changing very independently, then maybe you have the same microbiome or not, who knows? Yeah. But at least from the pouches of different marsupials that have babies, then at least there is a little hint there that even though, these are both marsupials, but even so, they are different and they don't talk to each other, they don't live together, but even so, they kept the same bacteria for their babies. So it will be very, very interesting to see for other functions and other things and other habitats, what's happening with the microbiome.
Yeah, exactly. And it's been a huge amount of time, 170 million years since marsupials and placentals shared a common ancestor. So have some microbes stuck with them the whole time? I don't know. Maybe there are a couple, maybe there's one microbe that has, or maybe there's a few. Or have they just picked up microbes from the environment, the right types of microbes for that particular task? So
yeah, well, that's your swab in the air. So so all this contamination might have been actually how they got in there in the first place.
Yeah.
Yeah, so that is super, super interesting. And we came full circle from convergence to convergence again. Thank you so much for the chat. I don't know if there's anything else you would like to add or you like people to know about your exciting work.
No, I think that's pretty much it. I just want to, say thanks to a few people. So obviously Anton Alberti, who's my current boss at the CEH. And then obviously I've got some people to thank as well. So Chris Helgen has been a real big, a big help in my career to date, and David Taggart as well. So I want to say big thanks to them.
Thank you very much for sitting down to have this chat at your 6 p.m., 8 a.m. here. This is how we do work nowadays in the world. It doesn't matter where you are. You can still work together and have a chat. And so technology is a great thing. And we can all learn from people living all over the world, which is super, super nice. And we are looking forward to having you in Copenhagen at some point when you join us all the way from Australia. And just one last question. So the Earth Hologenome Initiative has people from all over the world collecting these different samples on so many species. And then Anton has developed or designed a very fancy, cute kit that Aoife sends out and then comes back with samples. So for the Convergence Hologenomics, are you going into the field, or do you have specific colleagues, or is it a little bit like the Earth Hologenome Initiative where people also will send you samples kind of from all over the world, but I guess you have some target species which is quite a lot looking at all these pictures. So it is a big endeavor.
Yeah. Yeah, no, so it's a bit of both actually. So I do get to do a bit of fieldwork, which is pretty fun. But then I'm also really relying on colleagues, experts who have, they're, the masters of that particular species. They've been studying that species for decades, right? So I do rely a lot on these colleagues that will, donate their time and efforts to help, collect some samples, which is really cool.
Yeah, it's really, really cool to see this kind of project where if it was just one person or one small group doing the research, then you would get a lot less data. Whereas if you do this kind of globally and with people and collaborators everywhere, then you can get so much more information and knowledge from those samples, and faster as well, because otherwise you would have to be super rich and have a lot of time to travel and go all over and do all this sampling.
Yeah, definitely. And then, just having that expertise from people who know about this animal, know the ins and outs of it, that can really help us interpreting how to catch a wombat for one. But then also, factors that could influence what we find in our data, like we can kind of springboard off them a bit and say, okay, why do you think this might be the case here? Or what's your interpretation on that? And, I think that's really important. Rather than just saying, we'll just take something and sequence it and try and find something out. I think it's really important to have that, expert input and collaboration.
Definitely. That is definitely the way to go nowadays, with collaboration and multidisciplinarity. Thank you so much again. And best of luck for this project, because I'm very curious to see the results, which is might be a long way away still.
Yeah, no, thanks. And thanks for having me on to talk, Anna. It's been really cool.